Hi, everyone. Today we will discuss the second research paper. This paper is a typical study on proteomics that we learned previously. This is a reminder slide showing you that proteome is the collection of proteins present in a cell at a given time under specific conditions. As the applications of proteomics, we discussed the research project in Dr. Mohan's lab at the UH. He applied the proteomics study to identify novel biomarkers from lupus patients for a diagnosis. You still remember? The title of this paper is Tissue-Based Map of the Human Proteome. This paper was published in Science Journal in early 2015. Although I have been using the website this paper reported, it is my first time actually reading this paper. There are several reasons that I choose this paper. First, it integrates at least 2 genomics studies that we learned before. Therefore, by studying this paper, I hope you not only learn the concept of genomics studies, but also learn how to translate them in applications. Okay. So second reason is that this paper uses a few very useful technologies, such as immunohistochemistry or an RNA-seq, okay, RNA sequencing. And the third point is the figures showed in this paper are very popular in genomics studies. They are not only the old school figures, they're not like the old school figures such as the Western blot analysis or those agarose gel pictures, okay? By studying this paper, I hope you get familiar with the display of these figures. It will be very useful if you read the research papers in the future, all right? So as I mentioned, this is a report of the Human Protein Atlas, or called the HPA project, which is a Swedish-based program started in 2003 with the aim to map all the human proteins in cells, tissues, and organs. This project integrates various omics technologies, including the antibody-based imaging, mass spectrometry-based proteomics, and transcriptomics and systems biology. Okay, all the data is open access to allow scientists both in academia and industry to freely access the data for exploration of the human proteome. Okay, on this slide, I listed the website of this database, and you can go there and take a look. Okay, so the PDF file I uploaded to Blackboard contains 2 articles from the same group. The first is a summary of this study. I feel like it's between a very long abstract or like a preview article, okay? And the second one is a detailed description of this study. I will only go through the detailed one, the second story, okay? Here shows resolving the molecular details. So this is the abstract, okay? In this abstract, the authors mention that one of the major techniques they have used is called immunohistochemistry, or abbreviated as IHC. Okay, I'm sure that many of you may have learned this technology in other classes, but we haven't talked in our class. Okay, so this is a technique that uses antibody to stain your protein of interest in cells or tissues. In the article, we will see how the IHC results look like. So also, while I'm preparing for this lecture, I found that YouTube actually has a very good video introducing this program. I would like to share with you before I go into the figure details. All right.
Deep down in us, we find what makes us function, what makes us human, our proteins. And now, we're seeing them like we've never seen them before. All humans have approximately 20,000 genes coding for proteins, which are responsible for all functions of life in the trillions of cells that constitute the human body. In our kidney cells, proteins that filter our blood are produced. In the brain, proteins aid the formation of our memories. In blood cells, immune-related proteins are made to combat infections. Understanding human proteins is essential for understanding human biology and disease, to comprehend human health, and to develop better treatments for illnesses. The Human Protein Atlas database is a resource encompassing all proteins in the human body, allowing researchers to explore the complex network of these molecules. And this unique resource is open to everybody.
Every day, thousands of scientists are using this information to explore the molecular universe of our bodies, gaining deeper insights in the involvement of proteins in human biology and diseases.
The HPA Consortium is headed from the Science for Life Laboratory, a research institution for the advancement of molecular biosciences in Sweden. SciLife Lab started out in 2010 as a joint effort between 4 universities in Stockholm and Uppsala. The center provides access to a variety of advanced infrastructures in life science, creating a unique environment for health and environmental research. Spearheading the HPA project is biotechnologist Matthias Ullén.
The Human Protein Atlas was created by scientists in Sweden together with scientists all over the world. The aim is to create a periodic table of the building blocks of human beings, the proteins.
The Human Protein Atlas consists of 6 different categories, each exploring different aspects of human proteins. The Tissue Atlas contains information about the location of proteins in all major organs in the body, such as the liver, heart, kidney, pancreas, and the brain. Millions of microscope images created by the HPA Consortium are available here, allowing the study of each protein down to the single-cell level. The Cellular Atlas focuses on the smallest unit in the body, the cell. Multiple cellular compartments within the cell are analyzed for the presence of a particular protein, including mitochondria, endoplasmic reticulum, Golgi apparatus, and nucleoli. The Pathology Atlas shows the effect of protein levels for the survival of patients with different cancers. The results are presented as survival plots, and scientists can explore the relationship between the levels of a particular protein in a tumor with the expected survival of the corresponding patient. The Blood Atlas shows information about the immune cells and the proteins present in the blood. Here, scientists can identify proteins which are restricted to a certain cell type, such as, for example, the regulatory T cells. The Brain Atlas shows the proteins localized in the different regions of the brain. A large number of regions of the brain have been analyzed and displayed not only for humans, but also for pigs and mice. This allows scientists to explore the presence or absence of a particular protein in the human brain, and also to identify differences in protein profiles between these 3 mammalian brains. The Metabolic Atlas explores the enzymes responsible for human biochemistry. A separate atlas has been created, and data from this mapping effort has been incorporated into the Tissue Atlas. Here, it's possible to study the enzymes not only in 2D, but also in 3D. For each protein, a page has been created to allow researchers to get a summary of the information gathered by the content of the different parts of the Human Protein Atlas. For example, the information for insulin involved in diabetes is shown here. The summary page also gives information about the function of the protein and if the protein is involved in any human disease. The search function on the home page can also be used to make simple queries like finding a protein by name, or more complex ones, like FDA-approved drug targets with data in the tissue atlas and with high expression in a certain cell type in the kidney. This gives us 93 hits, with the third hit being the receptor for COVID-19. The Human Protein Atlas also contains numerous tutorial pages and a dictionary to guide researchers interested in a particular part of the atlas.
All the information in the Protein Atlas is provided without restrictions to allow scientists to get a holistic map of the human body. We are delighted that scientists from hundreds of countries are using the information from the Human Protein Atlas to facilitate the development of new diagnostics and new therapies to benefit mankind.
When we explore the world in new ways, we discover new things about us, about life itself.
Okay, that's a nice video, right? So this is figure 1. In this figure, the authors gave a summary of the tissue samples that they used in this study. If you recall the central dogma I always emphasize in this class, this study focuses on 2 kinds of molecules, RNA and protein. RNA was characterized as transcriptome, and the protein was characterized by IHC. So showing in this figure A, the authors showed which tissue RNA was analyzed and which tissue protein were analyzed. Okay, and then in figure B, the heat map shows the correlation between all 32 tissues based on the transcript or RNA expression levels. So how to read this heat map? Okay, first let's see the legend showing on this left upper corner. The higher the correlation, the more similar of gene expression pattern between 2 different tissues. For example, okay, say the testis has an expression of gene A, B, and CD. Then the liver correlated. This is the testis, and then this is the liver. Okay, so say the liver may have an expression of gene D, E, F, and G. Therefore, testis and liver has low correlation on the genes they expressed, because the only commonly expressed gene in these 2 tissues is D, gene D. Okay, but on the other hand, say colon may have expression of genes like W, X, Y, Z, and the rectum, shown on the bottom, may have a gene like XYZ. Therefore, colon and the rectum, they are highly correlated because both of them express gene X, Y, and Z. Okay, so in the map, then you will see this dark red color. That means indicates they have a high correlation. Of course, this is just an example. In this heat map, the investigators included 20,334 genes instead of just the 3 to 4 genes I mentioned as an example. Okay. So figure 1C shows the number of genes classified in each expression category. So the criteria of tissue enhanced, group enriched, and tissue enriched are listed in this table 1. I'll show next in the next slides. So figure D here is a Venn diagram. This is circle that overlap each other. they call the Venn diagram, okay? This Venn diagram showing the overlap between protein evidence on the basis of 3 online sources. One is the Human Protein Atlas in this study, showing this light blue. And then another one is Uniprot and called Proteogenomics. So the other two are also online databases. You can see that all 3 databases are heavily overlapped. The figure 1E and 1F are some other classifications based on the numbers of detectable genes at RNA or protein levels. Those results are a bit confusing, therefore I just skip them, okay? They're not going to be included in the exam either. Okay, so this table is another way to show the same data as figure 1C, but with additional information on how they define tissue enriched, group enriched, and tissue enhanced groups. Please remember this classification was based on transcriptome from RNA-seq or RNA sequencing. So I hope you can still remember how RNA sequencing works. Here, the authors used the FPKM as a criteria for the level or amount of RNA in RNA sequencing. Then what is FPKM? So it stands for these fragments per kilobase of transcript per million mapped reads. Okay, so this is a quantitative way that the next generation sequencing machine reads the seed in the library during RNA sequencing. So just remember that the higher value of FPKM, the more abundant the RNA is. Okay, so you can see that 44% of the genes they measured are detected in all tissues, indicating these are housekeeping genes. So housekeeping genes are the genes essential for cells to survive. Some examples are like cell skeleton genes and metabolic genes. Interestingly, 9% here, 9% of the genes are not detected in any of the tissues, indicating that it is possible that some genes in our body are never expressed, or if the tissue samples the investigators used were or adult samples, it is possible that these genes are only expressed during development, but not in the grown-ups. So I actually try to find out information on the biosamples in method section in the supplemental materials from this paper, but the description is very vague. I did not find any information on the ages and or where they got those biosamples. Okay, here comes figure 2. So in figure 2, from A to N, they show the tissue expression and the localization for a selection of human proteins. So this is at the protein level. So on the top, these blue bars, the columns showing here, are the mRNA levels of this gene in different tissues. All right, and the bottom tissue pictures, these are the IHC results, okay, which detecting protein molecules. The brown colored stains, those show very brown, those are antibody staining. Basically, they show you the localization of the protein you are interested. For example, is this in the cytoplasm? Is this in the nuclei? Or this in some other places? And also it shows how abundant they are. Usually the darker the brown color suggests that the higher expression of the protein, okay? So the purple and the blue staining, also showing over there, those are H and E staining. That indicates the morphology of the tissue and cells. You can see by this H and E staining. So if you say you are a pathologist or if you learn the histology very well, you could immediately identify what tissue this is by just looking at the H and E staining, okay? Then what is tissue microarray? Because the title here is tissue microarray based protein expression. Okay, so sometimes it's also called a tissue array. Basically, it is a glass slide with many small pieces of tissues sticking on the slide. So when you perform IHC staining, you could stain all the tissue pieces on this slide simultaneously. Okay, so you can see that in this project, it involves a huge amount of staining work. Therefore, tissue microarray can help the investigator to generate results very efficiently, because one staining, you basically stain multiple tissues, right? Then in the figure 2P shown here, it shows the pathway analysis of tissue elevated genes in 13 selected tissues or groups of tissues. There are many different bioinformatics methods to analyze cellular pathways based on gene list. So here the investigators used called a GO or GO analysis, which known as gene ontology analysis. So this is a computer program that predicts which biological function or pathways the genes are involved, just based on the gene list that you provided to the program. Okay, not surprising, for example, in this blood and the immune system, this GO analysis related the gene function to immune response and the cell activation. All right. And then here shows this figure 2.0 has the number of elevated genes in these 13 organ systems shown in P. So these are very self-explained, therefore I will not go into the details. So with so much data the investigators have in hand, they further classified these genes based on their cellular localization. All right, so shown here is in figure 3A, all these genes, this actually the N number is more than 20,000. They are classified into the categories as soluble, membrane spanning, and secreted, as well as genes with isoforms belonging to 2 or all 3 categories. How did you define where the gene's product is located? You may want to find this information in the paper, okay? And then figure 3B is again Venn diagram. They're showing the numbers of genes in each of the 3 main subcellular localization categories, like membrane, secreted, and soluble. Okay, the overlapped genes are isoforms that belong to 2 or all 3 categories. Then what are isoforms? So isoforms are any of several different forms of the same protein arising from either SNP, alternative splicing of mRNA, or post-translational modifications. We have learned all this before, right? And then in later in next few more slides, I'm going to show how the isoform will look like, okay? So again, there is another way that investigators show the same data. So in figure 3C, these genes are now classified based on the criteria shown in table 1, we just talked about. So in figure 3D, they are shown by different tissues. You may notice that these kinds of graphs give you 3 groups of information. One is the x-axis, y-axis, and then different colors, right? So with the availability of the big data sets, there is a trend to use one graph to show as much information as possible. So we will see an example with 4 groups of information in one single figure next. So these are the actual 3 characters: tissue, and the fraction of transcripts, and also here shows where they're located. So in figure 3E here, The investigators selected the 370 most abundant genes. That means its FPKM value more than 1,000. So these are from different tissues, and their predicted cellular localization is shown by different color. They have a small legend here. The gene names for a selection of the most abundant genes were labeled. All these different genes, you can tell the name, right? Therefore, 4 factors are displayed simultaneously in this figure. One is tissue, and then you show this mRNA level on the y-axis, and cell localization, as well as the gene names, right? So from this figure, it is obvious to see that most expressed genes are secreted, right? And the pancreas has the most secreted proteins in the tissues examined in this figure. Similarly, in figure 3F, the MR level of all genes identified as tissue enriched are shown for a few selected tissues shown on the bottom. So here, each dot stands for one gene. Therefore, the factors shown in this figure are tissue showing the x-axis and mRNA level showing in the y-axis, and cellular localization is showing by the different colors, and gene shown as the different dots. So also 4 different factors, right? So one striking result you can immediately see is that testis has the most numbers of expressed genes. There's so many dots, right? And the majority of these genes encode soluble proteins because you see this color majority, they are soluble genes, soluble proteins. So brain has the second most number of expressed genes, but majority of these genes encode the membrane proteins because there's a pink color. So please make sure that you understand and know how to read these figures, okay? Now let's see figure 4. And I will only go through figure 4F and figure 4G, okay? The whole figure is trying to correlate the transcriptome with cancer target genes. And I found these 2 figures are interesting. So we'll not test the other figures, other like sub-figures in this figure 4. So these 2 figures are pairwise comparison of mRNA levels between normal tissue and cancer cell lines, okay? Each dot here represents one gene, similar to the previous figure, okay? So figure 4F is a comparison between this normal liver tissue and a liver cancer cell line named HapG2, the HapG2 here. Okay, similarly for G here is a comparison between pancreas normal tissue and a pancreas cancer cell line named KPEN2, okay? So for instance, in G, you can see the red dots here, which refer to tissue enriched genes, showing here. And then they are highly expressed in normal pancreas because all this reading, the normal are really, really high, right? But they are very low. When you see the y-axis, they're very low, or even loss expression is not almost not detected in the cancer cell line. So what does this mean? So please try to find the answer in the paper, okay? This is important. Okay, now let's go to figure 5. This is actually last slide I'm going to show. So in figure 5, the investigators characterized the tissue expression of gene isoforms. I selected the figure D and E for an explanation. Other figures will not be tested, okay? As I just mentioned, many genes make different isoforms at mRNA level. Showing in figure 5D here, this gene named LYNX1 that has 9 different isoforms. these isoforms have different composition of exons, right? So the isoforms labeled with pink, 3 of them, they are predicted membrane spanning isoforms, and meaning that they are membrane proteins. But whereas the blue colored isoforms, these are secreted, predicted secreted isoforms. So I hope you still can identify exons, and then these introns, and then these UTR, showing this gray here, because we learned and tested all these before. Maybe I will test it again, okay? So on the bottom here, which we haven't learned, okay? So these colorful peaks, what are they? So these are RNA-seq reads, okay? So it actually skipped this y-axis here. It didn't show y-axis, but which is supposed to be FPKM, I believe. So the peaks here are the levels of mRNA expression in their corresponding regions. The regions actually showing from here to here, right? And then you may notice that no peak is detected in the intron level. For instance, the intron somewhere here in the middle here, it's empty, all right? Depending which isoform are expressed in different tissues, the peaks show the different shapes, okay? In brain and heart muscle, So this gene is likely express membrane-bound isoforms, but not secreted isoforms. Why? You should understand this, okay? Because the RNA reads spread through this very long 5' UTR region. This is only exists in membrane spanning isoforms. So one is 003, another one is 201. Both are pink, and then they are membrane spanning isoform. But in contrast, there's no reads found out in this region. So this is the first 2 to 3 exons that only located in the secreted isoforms. So that's clear, right? Okay, so also in figure 5E, showing here, the investigator shows the expression of 3 isoforms of LYNX1 in 32 tissues in this study, okay? So the secreted isoforms here, this is secreted, is expressed at high levels in what? The pink is esophagus, and the blue one is in skin, right? And then whereas the 2 membrane spanning isoform, one is 003 and 201, so both are the membrane bound form, they have high levels in the esophagus. I'm sorry, this is the yellow one is other tissues. And then this green one is heart muscle. So, no, I'm sorry, this is actually brain. I become color blinded. So this is yellow color, so they are highly expressed in brain and the heart muscles, both form. That is consistent with what we observed in this figure, all right? So then I hope you need to understand all the figures after I introduce this to you, okay? So there's an additional figure in this paper, figure 6. So it is about tissue-based map of human metabolism, because that is not related to what we have learned in this course. I'll just skip figure 6. And then of course, figure 6 will not be included in the exam, all right? So because the goal or objective of this paper is to introduce and present a database to the readers. So the figures are very descriptive. So you can see they organize in like different ways. So reading this paper, I hope you will get to know how to study and organize the figures for the omics research. And then I'm pretty sure like in the future, you will read a lot of similar articles to these figures. All right, so that's all for today.